To maintain biological order, human cells must continuously clear out their own structural waste. Two primary systems handle this: macroautophagy, which clears misfolded proteins, and mitophagy, which targets failing mitochondria for degradation. The upstream initiator for both of these clearance pathways is the ULK1 protein complex. When cellular components become damaged, ULK1 acts as the primary signaling node, directing the cell to isolate and destroy the debris. In Alzheimer's disease, this clearance pathway demonstrably fails, allowing toxic proteins to accumulate. However, the exact baseline expression of ULK1 in the human brain and how it fluctuates over time lacked rigorous clinical mapping. To establish this baseline, researchers analyzed serum and cerebrospinal fluid from a 391-person clinical cohort. The objective was to track exact ULK1 concentrations across normal aging, as well as through varying stages of cognitive impairment. The longitudinal data revealed a distinct trend. Even in cognitively unimpaired individuals, ULK1 protein levels dropped by nearly 20% in cerebral spinal fluid over an observed 4-year period. The aging process naturally depresses ULK1 production. This steady decline creates a baseline vulnerability, slowly compromising the brain's capacity to clear the very waste products that define neurodegeneration. That baseline vulnerability is highly pronounced in patient cohorts actively presenting with Alzheimer's pathology. These scatter plots track ULK1 concentrations across 3 groups: cognitively unimpaired controls, mild cognitive impairment, and severe Alzheimer's dementia. Look at the AD dementia column on the far right. The measurements show a stark reduction. Concentrations are drastically depleted compared to the control population. The researchers then tracked the rate of cognitive decline over several years, using the clinical dementia rating sum of boxes methodology. The results established a clear, predictive correlation. Patients who entered the study with higher baseline cerebrospinal fluid ULK1 levels experienced a demonstrably slower rate of clinical progression towards severe dementia. ULK1 reduction is a driver of disease progression, rather than a mere byproduct. This epidemiological correlation sets up a specific biological hypothesis. If falling ULK1 accelerates Alzheimer's, upregulating it should halt the damage. To test this mechanism mechanically, researchers engineered a transgenic mouse model designed to ubiquitously overexpress the ULK1 protein. They harvested primary cortical neurons from both wild-type mice and the overexpressing model to evaluate how well each isolated group withstood extreme biological stress. These cells were exposed to a panel of neurotoxicants. This included kinesic acid and NMDA to trigger severe excitotoxic stress, as well as rotenone to force immediate mitochondrial collapse. These dose-response curves map cell death. Across all 5 trials, wild-type neurons on top rapidly undergo apoptosis, while ULK1 overexpressing neurons display robust survival. A green tunnel assay confirms this. The wild-type row shows dense structural failure, while the overexpressing row remains clear. Artificially upregulating this single signaling node grants isolated neurons generalized structural resilience against catastrophic chemical environments. While general resilience is a strong indicator, Alzheimer's requires a highly specific defense. The researchers next exposed the cultured neurons directly to oligomeric amyloid-beta. This illustration details the Alzheimer's cascade. External amyloid-beta plaques induce intracellular tau proteins to misfold into rigid tangles within the somodendritic compartment. Notice the call-out box. As these tau tangles form, they physically disintegrate the internal microtubule network, destroying the cell's structural integrity. Exposure to amyloid-beta typically induces severe axon shortening. However, overexpressing ULK1 successfully counteracted this morphological damage, allowing the neurons to preserve their physical reach. Most critically, the overexpressing neurons exhibited a 24% reduction in the formation of somodendritic tau tangles compared to the wild-type controls. ULK1 actively severs the pathological link that translates extracellular plaque buildup into intracellular tau-induced neuronal death. To determine if this cellular defense translates to a complete mammalian system, the study utilized the 5XAFD transgenic mouse line. These mice are genetically engineered to produce massive, aggressive amyloid plaques early in life. Researchers crossbred them with the ULK1 overexpressing line to see if elevated clearance could overpower systemic pathology. By 7 months of age, standard 5X-FAD mice display severe spatial memory deficits, an impairment quantified using the Morris-Watermaze spatial tracking test. These circulant tracking diagrams record the animal's swimming trajectories. The diseased mice display chaotic search patterns. But look at the crossbred group in the bottom right quadrant. They execute a clean, highly directed path toward the target zone, proving clear spatial retention. The resilience observed in isolated cells scales directly to the whole organism. Elevating ULK1 protects systems-level cognition and memory retention in a living brain. This cognitive recovery requires physical proof. We need to know if the brain is actively clearing the amyloid plaques or merely surviving alongside them. This microscopy data provides the precise histological mechanism. Amyloid plaques are stained green, and microglial immune cells are marked in red. The overexpressing mice show a dense, targeted clustering of these glial cells directly around the remaining plaques. Beyond protecting individual neurons, ULK1 elevation significantly boosts the phagocytic uptake capacity of the brain's microglia. This turns glial cells into highly efficient biological scavengers capable of rapidly engulfing the amyloid. As a direct result of this efficient clearance, the surrounding tissue experiences a sharp drop in reactive astrogliosis, calming the overall inflammatory state of the brain. Age naturally throttles ULK1, causing an autophagic bottleneck. But by therapeutically targeting and upregulating this single pathway, we can clear plaques, block tau aggregation, and rescue memory. This firmly establishes the ULK1 axis as a primary target for future Alzheimer's drug development.